We have come to the last part or the last element, which is the sixth element on the post-transcriptional regulation, which is regulation of translation. So even though we're talking about post-transcriptional regulation, so regulation of translation is closely related to, well, if your protein is defective or something not right about it, so probably will not get translated. So hence we have regulation of translation. So translational regulation would provide a rapid mechanism to control your gene expression, and of course other numerous regulatory proteins which target the initiation step. Okay, and of course, okay, this is sometimes, okay, or often in a way that couples translation to mRNA localization. So the translational control in eukaryotic cells is critical for gene regulation during especially nutrient deprivation and stress, developmental and differentiation, nervous system function, especially in mammals, aging, and diseases. So this is usually achieved using proteins that bind to mRNA. So how does it do? Okay, inhibit translation by blocking mechanism and also extension via protection from degradation. So if it were to block, okay, so block would be initiated, okay, from the translation stage. So there are regulatory proteins, okay, that is attached to the 5' end of your mRNA, and this would prevent attachment of ribosomal subunits and initiator tRNA. So very similar to other mechanisms that you have seen before, it would block translation of mRNA to protein. Okay, so there is something, okay, in front of it, so it will not move on. So mechanism of translational control, basically transport control, okay, of only the mRNA which is transported, okay, to the cytoplasm can be translated, and this would more or less make sure that it is ready for translation. So it also involves number of ribosomes, translational factors, and of course if the concentration of this factor is too low in the cell, so translation start, okay, or the elongation process can be decelerated or inhibited. Next is mRNA localization, whereby there should be a specific place in the cytoplasm that leads to the production of the protein at the specific position itself, and regulation by untranslated region, your UTR. So overall, okay, this is what happens, okay, from transcription. Okay, so well, your DNA double-stranded, okay, is basically being unwound, okay, or melted, okay, the bubble being melted. Your polymerase 2, okay, would initiate transcription. You get your pre-mRNA with your exon and introns, okay. So later on, okay, they are processed. Okay, we know that, okay, cap is added, okay, splice, and also polyylation occurs. Okay, so you have your cap there, you have your poly A tail there, and you have your exons all joined together. Then they need to cross from the nucleus to the cytoplasm for the translation. So we know that from here to here it involves, okay, a nuclear export transfer, and involve lots of other mechanisms as we have seen in earlier videos. So for this to occur, again, probably you would know that, okay, some mRNA would go through some decaying processes. Okay? Some of them would go through stabilization, and localization, as mentioned previously, okay, is basically very crucial to determine translational regulation. Okay? So apart from that, okay, sometimes, okay, there is an RNAi pathway, gene slicing by small nc RNA. Okay, so this sometimes, okay, would determine how the genes behave and the protein to be translated. Okay, so it also basically tells you of other RBP functions. Alright, so again, because we are only focusing on mechanism and regulation, okay, of gene expression, so I'm not going to go too much on the translation, which is going to be more complex. So let's just have a look, okay, at different types of control, a bit of the description and examples. So we start off with transcription. I'm not putting in replication because, well, it's actually very easy. So transcription, okay, basically we know regulates, okay, which genes are going to be transcribed, and okay, the examples are access to promoters, activators or repressor protein, and methylation. So for post-transcriptional, it controls the availability of mRNA to the ribosomes, and this would involve alternative splicing, masking protein degradation, and a few more. So in the next is when you get your protein, okay, your mRNA to be translated, so you get your protein. So it controls how fast mRNA is translated to protein. So how fast or how slow is actually also crucial, because certain protein needs to be made at just the right moment of time, and sometimes they need to be expressed constitutively. So this example is actually in the variation of the poly A tail. So for post-translational, so it controls when and how long proteins will be functional, okay, because after which, okay, then sometimes the protein will cease to function or it will be degraded. So it involves, for example, okay, in the processing, okay, usually chemically modified or tagged for degradation. So there you have it, okay, everything about post-transcriptional regulation.